Good morning, students. I am Dr. Madhuramozhi Govindarajalu, HOD of Zoology. Today, I am going to talk about the transgenic animals. And the previous classes, I taught you about the genetic engineering, isn't it? That is the basic principle of genetic engineering. And taking this genetic engineering, how we are, this is nothing but the modification of the gene and how we can do the modification by transferring the gene. And there are various steps are involved in the genetic engineering. You know already the uses as well as the application of genetic engineering. So coming to the transgenic animal, this is nothing but the product of the genetic engineering. How we are going to transfer the gene. So if a gene is diseased or if a gene is under treatment, we need to change the gene that we want to keep a desired gene. Multiple copies of the gene we need, isn't it? So we are that for that purpose only we are going for the genetic engineering. So gene is dictating the type of protein that our body requires. So the gene is transmitting the information through the genetic engineering and also the protein synthesis, which is the main product of the genetic engineering. For example, we already seen how the insulin secreting gene is through the genetic engineering, we can produce large number of insulin secretion and how it is used for the diabetic patient. So by means of rDNA technology, we can do the genetic engineering. That is nothing but the recombinant DNA technology. So here also in the transgenic animal, this is the one that carries the foreign gene that has been deliberately inserted into its genome. How can we do deliberately? The selected gene, the foreign gene, that is the desired gene, is first inserted into the plasmid. They are nothing but the extra chromosomal DNA. They are seen other region of the other than the chromosome. That is, it may be in the cytoplasmic region. So these plasmids are the carrier, or this is otherwise called as the vector, and by which they are carrying the gene, the desired gene, for the purpose of genetic engineering. The transgenesis is a process by which mixing up of the gene takes place. We can mix the gene. So foreign genes are inserted into the germ line of the animal. So it can be transmitted because you already know any character which is transmitted through the germ cell which will can express, isn't it, which will can take the character, but those genes which are in the somatic cell, they cannot be carried to the next generation. So the transgenic technology has led to the development of fishes, livestock and other animals with the altered genetic profiles which are useful to mankind. Because our need only, we are going for the transgenic technology. We can get more amount of flesh in the case of fishes if we are doing the transgenesis in the fish, or we can do in the sheep and the other plant, everything, any animal we can choose for the transgenesis. So what are the production of the transgenic animal? How we can do this is coming to the methodology. First step is the construction of the transgene. So the transgene is made up of three parts. They are the promoter and the gene to be expressed and the termination sequence. Because when we are talking about the various tools involved in the genetic engineering, we expressed about the various enzymes are also, isn't it? Because enzymes are very, very important for the conversion of the one substance into another product. Likewise, the three parts are involved here: the promoter, then the gene to be expressed, then the termination sequence. Because after one such a period, we want to terminate, we want to end up the chain reaction. So if the polymerase chain reaction is continuously going on, it cannot make a point, isn't it? So we have to terminate. Then step 2 is the introduction of foreign gene into the animal. The pronuclear microinjection method we can choose, or embryonic stem cell method. Any method we can opt. So the microinjection method we can do by female animal is super ovulated, and they are we can collect the egg. The female animal is super ovulated, then we can get the eggs from the super ovulated animal. Then eggs are fertilized in vitro. In vitro method we can fertilize the that means in vitro in vivo. The in vitro means we are containing the eggs, the fertilized into a vitro medium. Then the transgene containing the solution is injecting. We are taking in the test tube, and then we are injecting into the male pronucleus using the micropipette. Here, this is mainly for the fertilization. So, egg with the transgenes are kept overnight in an incubator. So, everything we are doing in vitro. In vitro means outside the animal in the test tube. So, the eggs with the transgenes are kept overnight in an incubator to develop into two cell stage, because when you take after fertilization, the eggs are undergoing cleavage, 2 cell, 4 cell, and then 8 cell, 16 cell, 32 cell stages. Then they are coming to the blastula, gastrula, then the organ formation, everything you know about the embryology, gastrulation, etc. So the eggs are then implanted into the uterus of the pseudo pregnant female. We are taking it to the pseudo pregnant female, that is the female which has been mated with a vasectomized male. That is, the vasectomized male means they cannot produce sperm. So we are taking the two cell stage to the pseudo pregnant female. Then, because this is a one method of transgene, then the second method is embryonic stem cell method. That is, the transgenic animals can be created by manipulating the embryonic stem cells. The embryonic stem cells are obtained from the blastocyst, that is the inner cell mass of the blastocyst. Blastula, which is having various cells, isn't it? They are externally covered by a cyst. So it is called blastocyst. Cyst means hard covering. So the transgene is incorporated into the embryonic stem cells by microinjection, then by retroviral method, then by electroporation. These are the different methods we can take the transgene for the embryonic study cells, embryonic stem cell method. So the transgenic stem cells are grown in vitro, then they are inserted into the blastocyst and implanted into the host uterus. That means inserted into the blastocyst. Then we are implanting. Implanting means we are taking and placing it into the host uterus to grow normally. After that, when they are reaching the host uterus, they will grow normally. Before that time, we have to take care of the microinjection method and retroviral method, then electroporation method. Here we are choosing the embryonic stem cell method, by which we can create or manipulate the embryonic stem cells. So this is the diagram of the blastocyst micro injection method. Then step 3 involves the screening for transgenic positive. Where we have done the process, we have to know whether we have completed positively the process. So the transgenic progenies are screened. We have to screen them by PCR. PCR is nothing but the polymerase chain reaction to examine the site of incorporation, where the gene is incorporated, or otherwise we can call where the gene is recombined with that of the already existing gene. So some transgenes may not be expressed if integrated into the transcriptionally inactive site. We already seen about the endonuclease enzyme, activity of the endonuclease, and they are cleaving or cut the plasmid, and that particular end should be a sticky one. Then only we are taking the foreign DNA. This DNA will be recombined in the particular sticky end. Is it clear? So some of the gene may not be integrated. So we have to screen them. We have to identify them. Then fourth step involves the further animal breeding is done to obtain the maximal expression. How we are the heterozygous offsprings, they are mated to form the homozygous strain. And the first method, the DNA with the embryonic stem cells, how we are doing the method of embryo transgenic. Then the select for cells which are expressing the desired gene. We are selecting the desired gene and then injecting the transformed embryonic stem cells into the inner cell mass, because I already told you they are having the blastocyst hard covering. We have to inject it through the micro injection into the inner cell mass. Then this can be implanted into the uterus of the rat, for example. We are taking the white rat and they are said to be the foster mother. And then they are producing the offsprings, test offsprings for presence of the gene. Whether the gene is present or not, we have to screen them. Then it should be mated with that of the heterozygous offspring if we want the homozygous transgenic strain. So there may be some problems regarding the transgenic animal, and this is the multiple insertion may occur. So too much protein may be inserted, because when we are injecting through the micro injection, there may be the possibility of multiple insertion, and insertion into an essential gene, that is the cause the lethality, and the insertion into the gene leading to gene silencing also. Sometimes they cannot express. So it is called gene silencing. Then the insertion into the different area can also affect the gene regulation. So we have to choose the particular site, only one site for the insertion of the foreign DNA into the genome. So these are the some process involved in the transgene. And what are the results we can obtain? Some examples of the transgenic animal, how we have conducted the transgenes, and also how we have resulted in the transgenic fishes. We can get the superfish with increased growth rate and size, growth hormone gene inserted into the fertilized egg. Then the transgenic salmon grows about 10 to 11 times faster than that of the normal fishes. If the fish is undergoing the transgenic process, we can get more than 10 to 11 times flush or the growth rate. Likewise, we can do the transgenesis in the glowfish also. That is the GM freshwater zebra fish. Danio herrero, this fish is the zebra fish, and this is having much value, and also they are very important fish. We can do the transgenesis and produce by integrating the fluorescent protein gene from the jellyfish into the embryo of the zebra fish, and thus by we can get more amount of protein, as well as they are producing the substances which are said to be the anti-carcinogenic effect. Then some of the mouse we can do for the transgenesis process, the Alzheimer's mouse. In the brain of Alzheimer's patient, the dead nerve cells are entangled in a protein called amyloid. Some patient, due to old age, they are having the Alzheimer disease. That means they will forget the things. So for this person, the protein which are entangled or they are covered by amyloids. So mouse made by introducing amyloid precursors gene into the fertilized egg of the mouse. We can make artificially this transgenesis by doing in the mouses. So onco mouse, they are the mouse model to study cancer. I already told you, no, how we can do the mutation in the cancer cells, normal cells of the mouse, and this is by means of insertion of the activated oncogenes. Oncogenes, the genes responsible for the production of cancer. If we are stimulating or activating these genes, normally they should produce the cancerous cells. So this is the oncomouse. Then smart mouse, they are the biological model engineered to overexpress the NR2B receptors in the synaptic pathway. So some of the mouse we can do the genetic engineering for the expression, and this makes the mice learn faster like juveniles throughout their lives. We are using for the experimental purposes, the white rats and all. Likewise, we can do the genetic engineering in the smart mouse also. We can produce smart mouses. Then by means of transgenic mouse, we can reduce the Alzheimer's disease and also the gene which are responsible for the cancer. We can activate them and we can they can produce more amount of cancerous cells. So these are the transgenic mouses. Then transgenesis can also done in pig, that is Enviro pig. The pigs have trouble filling digesting a compound known as phytate. They are found in many cereal grains used in the feed them. So transgenic pigs are created by introducing phytase gene of E. coli. From the Escherichia coli, we can get the gene and inserting this gene into the pig. So naturally, the phytase enzyme is thus produced in the salivary gland of the pig, and it degrades the indigestible phytate with the release of the phosphate that is readily digested by the pig. Already they are not having the power to digest. Now we are doing genetic engineering by placing the gene which have the capacity to digest from the E. coli. So now they are converted into Enviro pig. Then pig for organ transplantation also. Pigs with human gene in order to decrease the chance of organ rejection by human body. So for example, if a patient is having failure of the kidney, doctors are advising for kidney transplantation, isn't it? So at the time, the patient body should adhere, they should have the capacity to absorb this foreign particle, that is others kidney also. But sometimes there may be the organ rejection occurs, isn't it? So the person cannot accept the transplantation, whether it may kidney or eye or any part of the body. Some people may not have the acceptance, so that only they are the immune system of a person is getting reduced, reducing the immunology of the person. Immune resistance is decreased, and because of that the foreign organ can be absorbed. And the transgenic in livestock, the bioreactor whose cells can be engineered to synthesize the marketable protein more economical and than the producing desired protein in the cell culture. So through the bioreactor also we can produce more protein. The transgenic cattle also created by the cows are made to produce protein, lactoferrin and interferons in their milk. And prion-free cows resistant to mad cow diseases, they are also developed. And transgenic sheep are also produced for good quality wool and meat. Transgenic goat, they could express tissue plasminogen activator, antithrombin 3, spider silk, etc. in the milk. Then transgenic rabbit can also be produced and created in 2000 as a transgenic artwork. Then transgenic monkey this was also produced during 2000, and an engineered virus was used to insert the harmless gene for green fluorescence protein into the ANDA's rhesus genome. So importance of transgenic animals are medicinal purpose, agricultural importance, and the industrial importance. And this is having very useful in the human applications and disease model, bioreactors for pharmaceutical industries, and xenotransplantation also done because of the transgenic animal. For agricultural purpose, we can produce disease-resistant animal for improving the quality and quantity of milk, meat, egg, and wool production. Then for industrial importance, toxicity sensitive transgenic animals to test the chemical. Then spider silk and silk of the goat can be produced. Then various problems, issues related with the transgenic technology are also existing. There may be health risk associated with the transgenics, and there may be long-term effect on the environment. When transgenic animals are released into the field, various bioethics argue that it is wrong to create animal that would suffer as a result of genetic alteration. So recent trend also in the transgenic goat engineered to produce human breast milk also They are also created. Then coming to the conclusion, transgenic technology is a field that is under constant evolution. Many transgenic animals have been successfully created for variety of purposes, and the prospects are enormous, and it holds great potential in many fields, including agriculture, medicine, and industry. So with proper research and careful use, the transgenic animals can go a long way in solving several problems for which the science does not have a solution till now. Thank you.